My name is Anson Rosenthal, and my project is modeling the propagation and geographical spread of Dracaena polymorpha in the Susquehanna River. A little bit of background. The zebra mussel is a freshwater mollusk native to the bodies of water in Eastern Europe and Asia. It was transported to the United States in the late 1980s through the ballast water and cargo ships, landing in the Great Lakes. And since then, it has spread really quickly throughout the eastern United States. Here's a picture, a little snapshot of the sightings in 1988. And 20 years later, it has spread through, again, most of the bodies of water in the northeastern United States in particular. And it's moving outward through both human vectors and downstream transport. Humans, they can carry the larvae called veligers in recreational vehicles or other vessels to other bodies of water that would not have been previously vulnerable. And in addition, there's the downstream transport in which the veligers are sent into the water column during the fertilization process, and they are swept downstream to a different area of that same body of water. Now, in addition to spreading rapidly, they cause two significant and imminent problems. They remove massive amounts of phytoplankton from the water column, and it is an observed 80% reduction in phytoplankton concentration in the Hudson River over a 2-year span. And they also block the water intake valves and circulation systems in both municipal water systems and industrial plants. So that poses a significant risk to the economics of industry on the rivers, particularly the Susquehanna River. So we have some methods of control I'll touch on right now. You have your physical methods, which are pretty clumsy and costly. I'll be looking at the chemical additives to the water to disrupt biological functions, which has been proven to be safe to not be harmful to the other organisms in the ecosystem, and pretty cost effective as well. So the objective is a plan for preventive action in the Susquehanna River, and I want to determine exactly specific treatments and the locations at which they should be implemented to best prevent a successful invasion. And the model I'm using is semi-discrete and spatially explicit. It's spatially explicit in that the river system of the Susquehanna is broken down into specific segments, and the locations joining these segments are known as nodes. And along these, at each of these nodes, I have data collected from the real physical river, describing the current velocity and the position in terms of the length of the river at that location. So I'll be using that to construct a virtual representation of the Susquehanna River, and it is semi-discrete in that there are two time scales running. In my model, there's the longer continuous time scale to reflect the progression and invasion over a span of years, and a smaller time scale to reflect the mortality and the settling of these veligers as they float along in the water column over the settling period, which is a matter of weeks. And you have the first component is the part of the model that will be operating at the spatially discrete level at one particular location, and it's a system of difference equations describing it breaks the population of adult mussels down into 4 age categories, and it relates their populations at a particular location X at the next year to those respective populations at that location at the previous year, and then their survival rates. So it's pretty simple. And you have your differential equations to describe the movement of these veligers downstream. First, there's a partial differential equation. These were derived by another group of researchers, and then they spatially discretize this PD into an ordinary differential equation, which I can use to evaluate the mortality and the survivorship of these veligers between particular nodes in my model, and it can be integrated much more simply with respect to the days that they've been in the water column. And first off, you have periodic and chaotic behavior that this model can take under different parameterizations. You have, with one particular setting of the survival rate of 4-year-old mussels under two different initial conditions, which are fairly similar in the grand scheme of things. You have very different long-term dynamics of the model. But when you have a different parameter value for the same parameter, you get the convergence to the same attractor, which is this periodic motion right there. And you can see that there's a pretty well-defined range of these chaotic and cyclical behaviors throughout variation of two different parameters. And parameter optimization is something that I did to optimize the model to describe the Susquehanna River as best as possible. I took data from the Hudson River, obtained from another researcher, Dr. Dave Strayer at the Carey Institute, and I compared the model predictions under certain parameterizations to this data over the years, and I used R-squared sum to assess the fit. And I then optimized the parameterization based on a minimization of the sum of the R-squareds using the Excel solver. And these are my final results. And then here's the optimized model in the long term. Again, it's a bit more complex, but it still exhibits a pretty periodic and predictable dynamic and contributing to possibly the stability of my model, which bodes well. Using Esri ArcView, this is the GIS portion of my project. I had to look into the USGS Enhanced Reach File 1.2, which is a compilation of data describing all the hydrologic systems in the United States, including a number of physical parameters, which I had to extract. I had to extract the mean velocity and distance of each segment along the Susquehanna River, which I had to select on my own through this process on the right. And then I color-coded the path that I selected from a model to be evaluated on per the mean velocity, because it's been shown that the villager survival is negatively impacted by the velocity of the river. So this could provide, in this orange and red area here, a natural barrier to their spread. And then this is the final, generally which kind of data I ended up from this GIS data extraction. I ended up with my node number, and then distance to the next node, and then the mean velocity along that reach. These are the key ones I'm going to be using in my mathematical implementation. Here's the first implementation, which I later realized was incorrect. But so again, I have two scenarios of Veliger introduction at the initial condition of the system. You have initial Veliger introduction, simulating, say, an accidental release, by the human transport, or continuous, simulating if a population had been established further upstream. And then you have this a sequential solving this ODE at each node, referencing the solution to the ODE at the previous node to solve each successive one. And in that way, I could solve the entire system of ODEs for every year and generate predictions as to the adult mussel density fluctuations over the long term. And this is kind of what it looked like, a time series over 100-year progression. You have the node, the node number representing the length of the stream, and the mussel density. It fluctuates pretty wildly, but it progresses at a pretty constant rate. Then using the model, I will, once I get it fixed, I will implement local scale increases of Veliger mortality to simulate the implementation of these chemical control methods, and these increase in mortality will be based on empirical results. And then this is the key factor that invalidated my previous implementation, was an oversight during the GIS data extraction. I misinterpreted two of the attributes. Length is actually in coverage units, which I took to mean the distance between the nodes, when really it was a parameter defined as SCGL, which is miles. So when I fixed that, I realized that the sequential method was not exactly valid, and it masked a fundamental error in my implementation, in that the actual ODE references the next node in addition to the previous node to describe not only the forward transport of Veligers due to the current velocity, but the backward transport of these Veligers due to diffusion. And for that, I needed the Veliger population at the next node at a given time, tau. So these equations had to be solved as a system of more or less simultaneously. So what I'll be doing is using an iterative method in which successive approximations will be taken so that the Veliger population density functions for each node will eventually converge to their true value via, again, I guess, sequentially solving the entire system and then reiterating the process, I guess. I mean, I haven't done it yet, so it's just an overview. But